Hello and welcome everyone. In this podcast, we're going to talk about some of the terms that we use when we discuss chromosomes and the genes that are located on them. I want to begin by talking about the difference between the terms chromosome and chromatin. These phrases are often used in almost an interchangeable way. However, they shouldn't be, because the two terms chromatin and chromosomes are actually quite distinct terms. So let's write them out here. Chromatin versus chromosome. I want to begin with how they are similar. They both describe a long stretch of DNA plus the proteins associated with them. DNA plus proteins. That's about where the similarities end. Chromatin is a very long strand of DNA. It's very difficult to see under the microscope. They can be, but it takes a higher magnification. And they will have proteins associated with them along their structure here. These proteins we're going to talk about in the next one or two podcasts, but let's go ahead and name them here. These are histone proteins. Chromosomes turn out to be a much more highly compacted structure of DNA plus proteins. And I'm just going to draw it like this. This is what the chromosome would look like before it's been copied, and then this is what the chromosome would look like after it's been copied. We can't see it in here as easily, but it's important to remember that this very compacted structure here in reality is just one long piece of DNA. But you can't really see it because it is so compressed upon itself, attached with various proteins pushing it together. Another difference is that chromatin is not paired, and chromosomes are paired. We haven't talked about mitosis or meiosis yet, but you might recall from an earlier class, perhaps introductory biology, that in eukaryotes, like humans, we have many chromosomes. And we have, say, chromosome 1, which we originally got from one of our parents, so we'll call it 1A. And then we have another chromosome that we got from our other parent, which we would call 1B. Chromosomes are always paired with each other. They're right next to each other. Chromatin, we certainly would still have chromosome 1A, and we would certainly still have chromosome 1B. Chromosome 1 from one parent, chromosome 1 from the other parent. However, there's no reason that they need to be paired. That is right. There's no reason that they have to be right next to each other. But chromosomes are. Chromatin, in part because they are not as compacted with proteins, they're much more open and exposed. They are metabolically active. Whereas chromosomes, largely because of their very compact nature, are metabolically inactive. And what we mean by metabolically active is that the DNA can be copied through replication. The DNA can be transcribed into RNA, and in general, the DNA is exposed so various enzymes and proteins can associate with it to copy it or to transcribe it. Metabolically inactive chromosomes no longer have their DNA exposed because of the proteins associated with them. And so they cannot be copied and they cannot be transcribed. I ran out of room on my whiteboard, and I did want to say one more thing about the differences between chromatin and chromosomes. So I'm going to erase this very bottom part here. So this next thing I want to talk about, when we compare chromatin with chromosomes, involves the cell cycle. We're going to talk about the cell cycle in much greater detail in the next few podcasts. But let me remind you of some of the basics of the cell cycle. Remember, you have a cell that enters the cell cycle in G1. It then moves into S phase, and then G2, and then it goes into the M phase, which could be meiosis or mitosis. We're going to talk about each of these stages in much greater detail. So the point I want to make here about chromatin and chromosomes related to the cell cycle is that chromatin is found throughout the cell cycle in G1, S, G2, and to some extent in M. So let's go ahead and write that over here. G1, S, G2, and to some extent in M. It's also found in this other part of the cell cycle that I didn't mention, and that's called G0. The reason we are in the chromatin state here is that during this part of the cell cycle, G1, S, G2, and G0, the DNA needs to be active. It needs to have its DNA exposed so that it can be transcribed or replicated. Chromosomes, however, are only found in the M phase, during mitosis or meiosis, when those chromosomes are pairing up with each other, as I show you here, pairing up with each other so that they can be segregated into new daughter cells. And again, we'll come back to this later, but I did want to point out while we're talking about it, that chromatin, the active part of the DNA structure, is found throughout the cell cycle, mainly in G1, S, G2, and I should write G0 here, and that chromosomes are exclusively found during mitosis or meiosis. Now, for the rest of the discussion for this podcast, we're going to focus on the chromosomes. And so I want to draw something similar to, as I just erased, this chromosome pair. So this might be what we might call chromosome 1A, that we got from one of our parents. And over here we're going to have the other one we got from our other parent, chromosome 1B. These chromosomes are nearly identical. The DNA that is packaged up in here, in fact we could kind of draw the DNA here, if that helps understanding that this is actually a piece of DNA here. These long pieces of DNA have nearly the exact same sequence. They contain the same genes and the same order of genes. Because of that, they can pair up nicely with each other during mitosis or meiosis. Because of the similarity, we would call these two pieces of DNA homologs. Now remember, I said these were all long pieces of DNA, and we said that a gene, on the previous podcast, that a gene was defined as being a piece of DNA that makes a product, like RNA, and eventually a protein. So these chromosomes here are made up of DNA, and because of that, they are the carriers of genes. So chromosomes are composed of many genes. So we may have a gene here, and it's going to be the exact same gene over here, because they are homologs. And we may call this gene A. This physical location of the genes, whichever genes we're talking about, but here we're talking about gene A. This physical location of gene A is called the locus of the gene. The locus is the physical location of a gene on the chromosome. Now remember, we're going to have multiple genes on this chromosome. This may be where we have gene B. but we're going to make it a lowercase b. Typically in genetics, when we use uppercase, that means it's a non-mutated version of the gene. There are no mutations in it. A lowercase means there is a mutation in it. And if we're using the same lowercase, that means it's the same mutation in each of these two genes. Again, this physical location of gene B in this case would be called the locus of gene B, the physical location on the chromosome of gene B. Finally, let's draw a third gene. Now obviously a chromosome has more than 3 genes. A chromosome could have thousands of genes on it. This time let's make it a capital C and a lowercase c to illustrate that the two different versions of these genes don't have to be the same. In this case they are big A, little b, but now we have big C, little c. We should define another term here, and that is the term allele. An allele could be defined as one of several versions of the same gene. These different versions are created by mutations. Different alleles result from mutations. These different alleles that form because of mutations results in variation. If we didn't have different alleles, we would have no variation. So again, alleles are simply just different versions of the same gene. So a good example here would be C. We have the same gene, C. It has the same function. Let's say the function is curly hair versus straight hair. They both code for a gene that's responsible for the shape of your hair. However, one of them is going to give you curly hair and one's going to give you straight hair. Same gene, but a mutation gives it a slightly different function in the pathway that leads to curly or straight hair. Finally, let's bring attention to a couple other terms here. When we have 2 genes, and remember any individual is going to have 2 genes. If these 2 genes are different alleles, such as we have here between big C and little c, so let's just write that. Big C, little c is an example of a heterozygous pair. Gene pair. Whereas big A is an example of a homozygous dominant gene pair. We call this dominant, we call it homozygous dominant. Homo means the same. Dominant means that they are both the unmutated alleles. Big A, so we call that dominant. Little b, and I'm running out of room here, but let me write it down. This is an example of a homozygous recessive gene pair. Again, homozygous, homo, because they're the same, little b, recessive, because they are the lowercase, meaning that they each have an allele that is mutated. So we say they are recessive. Heterozygous, to compare them to the homozygous versions. Heterozygous, hetero, meaning different, always have different alleles. In this case, it's an unmutated allele with a mutated allele. Okay, I'm going to erase this real quick here. Eukaryotic chromosomes can be classified as one of 4 types of chromosomes. So there are 4 chromosome classifications. And they are all based upon the position of the centromere. Okay, we haven't talked about what the centromere is yet, but that is a place on the chromosome. In fact, let's go ahead and draw it. Here's our chromosome. And a centromere is the place on that chromosome that is important for the cell cycle. This is the place where spindle fibers attach to pull those chromosomes apart so that each of the resulting cells have the same number of chromosomes. We'll talk more about that in a few podcasts, but just for now, let's look at this centromere here as the physical marker to help us classify what kind of chromosome we're looking at. In this example here, the centromere, shown here in the circle, is right in the middle of the chromosome. If you look at the whole stretch of the chromosome, it's right here in the middle. So we call that metacentric. Meta meaning middle. So it's a metacentric centromere. The next example, I'm going to draw the chromosome roughly the same length here, is called submetacentric. In this example, the centromere is not right in the middle. It's not at the end, but it's in between the middle and the end. So we're going to draw it, not here in the middle, not at the end, but somewhere in the middle, like right here. So we call that a submetacentric. The next example is going to be called acrocentric. And that is when you have your chromosome here, and that centromere is near the top, but there's still some DNA above it. It's not in the middle, it's not in between the middle and the end, but rather it's very close to the end, but there is still some DNA on the top. But most of the DNA is on the other end of the centromere. But the important distinction is that there is some DNA at the top. The last example is going to be telocentric. Chromosome. And in this case, the centromere is all the way at the top. To distinguish it from the acrocentric chromosome, the telocentric chromosome has no DNA on one end of the centromere. All the DNA is on one end of the centromere. Now, there is no rhyme or reason as to how each of these have evolved, meaning that there's no known advantage to having the centromere in the middle, somewhere in between, such as the submetacentric, or all the way at the end, or near the end. These result throughout evolution for various reasons. None of these locations appear to have a distinct advantage that we know of. And I should also point out that not all eukaryotes have all of these types of chromosomes. For instance, humans have no telocentric chromosomes, whereas mice, the standard house mouse, only have telocentric chromosomes. Now we're almost done with what we want to cover in this podcast, talking about terms associated with chromosomes, but I want to move to a PowerPoint slide now to describe what a karyotype is and the different things we can gain from looking at a karyotype. Okay, for this last part of this podcast, I want to talk to you about what a karyotype is and how we look at karyotypes and what information we can get from a karyotype. A karyotype is simply a micrograph image of all the chromosomes that are found in a single cell. And then these chromosomes are arranged in a manner where you can compare similar chromosomes. So what happens is that you will crack open a cell, somebody who makes these karyotypes, they will break open cells, and what they initially see is something like this, where you have this collection of all these chromosomes from a single cell, just kind of spread all over the place. And so what the geneticists will do is they will start to find chromosomes of similar length and similar banding. If you'll notice, each of these chromosomes have these dark bands on them. And all the chromosome 1s are going to have the same banding pattern. And all chromosomes all chromosome 1s will have the same length. And through this process, you'll eventually get something that looks like this, where all the chromosome 1s are lined next to each other based upon their length and their banding patterns, as you can see here. All chromosome 2s, both of them that is, are lined up with each other. Same with 3s and 4s and 5s, and all the way down to chromosomes 22, and then the sex chromosomes Y and X. You'll notice as we're looking at these that chromosome 1 is always the largest chromosome, and then each chromosome gets successively smaller until you get down to chromosomes 22, which are the smallest chromosomes. That means that chromosome 1 likely has many more genes on it than chromosome 22. When a geneticist looks at these, he or she may want to look to see if there's any abnormalities. What we're looking at here is what appears to be a fairly healthy karyotype. The chromosomes are each the same length when you look at a homolog pair. So again, remember, we called these on the drawing before homologs. We also notice that there are not any extra chromosomes. For instance, we don't see an extra chromosome 8 here. We see the same number of chromosomes. We also don't see any chromosomes missing. We don't see only one chromosome 8 or only one chromosome 14. We'll talk in a few podcasts from now what these abnormalities are called when we're missing one or more chromosomes or have additional chromosomes. In some cases, we'll find chromosomes that are lacking a piece of the chromosome. So we'll talk about one specifically that's missing a piece of chromosome 5. This particular chromosome 5 is fine, but if you're missing a small piece here, it can lead to a very distinct chromosomal abnormality. So this is what we're looking at here is a fairly standard healthy karyotype. We don't see anything abnormal about it. A couple of other terms we should point out here is that chromosomes 1 through 22 in humans are called autosomes. Other organisms will have different numbers of chromosomes, but all the chromosomes that are not sex chromosomes, that is, chromosomes that are important in determining the gender of the individual, would be called autosomes. Whereas the other two chromosomes in this case are called sex chromosomes. A karyotype from a male will contain one Y chromosome and one X chromosome. If this happened to be a female, it would contain two X chromosomes and no Y chromosome. Again, we'll talk about some chromosomal abnormalities where there are different levels of these two chromosomes, the X and Y. But what we're looking at here is a fairly healthy chromosome containing one Y and one X chromosome, so we could say this individual is very likely male. We'll talk about a situation later on where somebody could be XY and actually be a female. But for now, if you see this kind of a karyotype on an exam, and I were to ask you what is the gender of the individual, you would definitely say this person is male. We'll save the exceptions for later. One other thing I want to point out to you, we said it on the drawing, but sometimes this point gets lost as we move throughout the course. Remember that each of these chromosomes, for instance, this one maybe came from the person's mother and this one maybe came from their father. Remember that each of these chromosomes is actually one large piece of DNA in a double helix. It's a humongous molecule, but it is still just one molecule of DNA for each of these chromosomes. Okay, I would like to define two more terms that we haven't had an opportunity to define yet, and these are diploid and haploid. Let's begin with diploid, because this particular karyotype we are looking at is an example of a diploid. In humans, all of our cells with the exception of sex cells, like sperm and egg, are diploids. So every cell in our body, except sperm or eggs, are diploids. Diploid means that those cells have 2 copies of every chromosome. So we have 2 copies of chromosome 1, 2 copies of 2, 3, 4, 5, all the way down to chromosome 22, and we have 2 sex chromosomes. In this case, it's Y and an X. So that's what a diploid is. Haploid cells are also found. We see haploids existing in different kinds of fungi and in sex cells of eukaryotes, such as sperm and eggs. Haploid cells only have 1 copy of each chromosome. So for humans, there would be 1 copy of every chromosome from chromosome 1 to 22, and then there would only be 1 sex chromosome. In this case, it's a Y. So if these are human sperm, they're only going to have either an X or a Y. If they were human eggs, then they would only have X chromosomes as their sex chromosome. So again, a diploid cell has 2 copies of every chromosome, and a haploid cell has only 1 copy of each chromosome. Okay, so that ends this podcast. Let's close with a quick summary. In this podcast, we talked about the differences between a chromosome and chromatin. We talked about how chromosomes and chromatin were the place where genes were located, and we talked about how alternate versions of genes caused by mutations were called alleles. And then we talked about the fact that the physical location of the genes on a chromosome was called that gene's locus. So you should be familiar with these 3 terms. We also talked about how chromosomes were paired up as homologs. That we get 1 chromosome from 1 set of chromosomes from our mother and the other set from our father, and that those sets combine, and chromosome 1 from each parent would be called homologs. We then talked about how gene pairs could either be heterozygous. I think the example we gave there is big C, little c, and how they could be homozygous, dominant. We gave the example of big A, or they could be homozygous recessive. And the example we gave there was little b, little b. We also talked about karyotypes. And how you could look at a karyotype and determine the relative health of the individual by looking at the number of chromosomes and the sizes of those chromosomes. We talked about the differences between autosomes and sex chromosomes on the karyotype, how we could determine if that karyotype came from a male or a female. And we also talked about the differences between diploids and haploids. So that is all I have for this particular podcast. If you have any questions at all, please let me know, and I'll help answer any questions you might have. I'll see you in class. Bye.